The first test that we're going to do is the student's t-test. Now, it's important to sort of really kind of appreciate and understand why and when this test is used. The student t-test is often called a means comparison test. And so basically, it's going to be utilized when I've got two data sets, two groups of data. And I want to assess, and it's going to be, I'm going to explain it again in the next slide with a bit of visual aid. I want to assess whether the averages, the means between those two sets of data are significantly different to each other, or whether the difference is due to chance. Let's see exactly what we mean by that. So for example, say that I'm comparing the average height of two different populations. So I have I set up an experiment and I want to ask, are people in the UK taller than people in Greece, for example? So I have two populations, two sets of data, and I figure out the average height in one population, the average height in the other population, and I want to see whether there's significant, is there an underlying biological reason why one population would be on average taller than the other population, or is it just due to chance? And this is what the t-test is going to tell me. So the difference in mean heights is what I'm measuring. My null hypothesis is that it's due to chance. And the alternative reason, the alternative hypothesis, is that there's an underlying biological reason. Now, let's break down this formula here, which is a little bit scary. But we're going to break it down. So the t-test value that I calculate depends on these factors. x1 and x2 is the average or mean of sample one, of my one population. And this is the average or sample or mean of sample two. Oh, come on. Sample two. What are S1 and S2? They stand for standard deviation. So it's basically standard deviation of sample one, standard deviation of sample two. What are the N's finally? They are the sample sizes. So how many individuals did I measure for height, for example? Sample size of sample one and sample two. This already tells me that the result of my statistical test, of my t-test, is going to depend on how big the populations are. If I'm asking the question, are people in the UK taller than people in Greece, and I only measure 10 people's heights, well, that's not going to really be a significant difference. It's not going to really help me for my experiment, for my question. Oops, sorry. So, what is the null hypothesis? As always, the null hypothesis states that there is no significant difference between the means of the two groups, right? And so, for example, I've got group A and group B, and I am looking at the average heights between them. So one is 35, one is 32, for example. So in order to actually reveal whether there's a significant difference between them, I need a large sample size, and also my data has to be what we call normally distributed. And these are the conditions that I need in order to be able to apply the t-test in the first place. If these conditions are not met, if I have a really small sample size and my data are not normally distributed, then I'm going to have to utilize a different statistical test. And you don't need to worry about that because you don't learn about those other tests. There are hundreds of different statistical tests that you can do. The degrees of freedom. So again, I'm going to have similar tables like I did with my chi-squared. I'm going to have to read off the degrees of freedom, and so on. And the degrees of freedom here is actually going to be determined by the size of the groups, by the n numbers that we had in the formula. I also need to have similar sample sizes in the two groups. I can't measure 10 individuals in one and 1,000 individuals in the other. Let's see an example, relatively simple example, straightforward, but a lot of work. So a new weight loss treatment for humans is being tested. The test group receives the treatment with every meal, while the control group receives a sugar placebo. Using the data below, determine if there's a significant difference between the control group and the test group. So some of the work has been done for me already here. I'm not given the raw data, and I don't have to calculate the average and standard deviation. However, I do still have to be able to apply it to the t-test formula, and the t-test formula will be given to you guys, as well as the chi-squared formula, so don't worry about that too much. Let's break down our question. So the context here are the data provided. The command, simple, is to determine the t-value. How? Subcontext, by comparing it to the critical value on the table. The values on the table, guys, whether it's t-test table or chi-squared table, we call them critical values because it's against those that I'm going to compare the value that I will calculate. And what is my direction? That I'm going to use a t-test. I'm being told that. I'm going to use a t-test. So take a minute to see whether you can apply these numbers here to the equation. One thing I'm going to say is that for the denominator, which is a little bit complicated, you will solve what's inside the square root first, and then finally apply the square root. Remember that x are the mean, s is the standard deviation, n is the sample size. So calculate it first, and then we're going to look at the table, the t-test table, and the tables are always given to you, of course. So let's plug in those numbers. The x's are this column here, the mean, okay? So t equals, let me make it a bit big here so you can see it, okay? 8.7 minus 7.9 over. So we said we're going to solve what's inside the square root first. Standard deviations are those ones here. So 5.3 squared over sample size 29. Plus 6.5 squared over 33. I solve all of that, it gives me 0 point, oops. Did I do some- 0.533. Okay, so that is my t value. So next bit is to compare my calculated t value to the critical t value in the table. However, before I do that, how will so I get 2 marks here for calculating the correct t value. It was a 6-marker question altogether, right? How will I calculate the correct degrees of freedom? The formula is the sample size for one sample plus the sample size for the other sample minus 2. So I go and look at the table with the sample sizes and I apply this calculation, I get degrees of freedom of 60. So I go down this column here. Here's the row that I'm supposed to be looking at. The degrees of freedom. I get another mark for doing the degrees of freedom correct. So the critical value at the 5% significance level. So you can quote the significance level with a decimal like 0.05 or as a percentage, which is 5% here. And the critical value is 2. I get a mark for quoting the critical value correctly. Therefore, since our value is less than 2, we cannot reject the null hypothesis. So the 2 groups are not significantly different. There's another mark, there's another mark. So the probability of getting results by chance is greater than 5%.